Hey guys, so we're looking at section 2.4 for today. Let me get my stuff kind of moved around here. So section 2.4 is the last section of chapter 2, which means that we're getting closer and closer to our chapter 2 exam, which is coming up here soon. Again, if you need help with this stuff, because I think this is probably the most difficult stuff we've done all year, be sure to reach out to me so that I can help you with that, right? So section 2.4, put down the heading for me, section 2.4, chemical reactions and enzymes. And what we're looking at here is a firefly, right? And how they light up in the evenings, which they do behaviorally for lots of different reasons, but that is an example of a chemical reaction. And so that's something we're going to look at here as one example of chemical reactions as they occur in living things. All right. So our objectives here for section 2.4, we have 3 of them. First one, explain what happens to chemical bonds during chemical reactions. Investigate how energy changes affect whether a chemical reaction will occur, and explain the role enzymes play in living things and what affects their function. And enzymes are going to be something that you're going to hear me talk about really from here on out through the end of the year. At least through quarter 3 when we look at DNA and production of proteins, because enzymes play a really important role. So what we're going to look at today is very much going to be built upon in the next several chapters and throughout the remainder of the year. So make sure that we've got a firm understanding of this, all right? Okay, so chemical reactions by definition, these are changing one set of chemicals into another set of chemicals. That's as simple as we can make it. By the way, up here where I have RXN in parentheses, that's just my shorthand for writing out the word reaction. So I put it there just to let you know that if you happen to see it on an assignment, on a test, on a quiz, on a PowerPoint like this, you know what I'm referring to, okay? So chemical reaction, or RXN, is a change in one chemicals into something else. So the chemicals or the elements or compounds that we change in any type of chemical reaction are what we refer to as reactants. These are those things that when we write a chemical equation, we're always going to put on the left-hand side. So over here on this side of my chemical equation here are where I have my reactants. Okay, so the reactants are what we always start with in any chemical reaction. What we're going to produce in a chemical reaction are what we call the products, and those are always written on the opposite side of the chemical equation. So I'll label this one here as products. And it would be a good idea if you also wrote down this chemical equation or something similar and labeled the reactants and the products as like a really kind of finite example of what we're looking for here. Okay, so reactants are always on the left, products are always on the right, and then we tend to have in the middle this arrow here. And this arrow in the middle is not representing flow of energy like we saw in food chains and food webs. This is going to be the direction of the reaction, right? So we're going to say reaction direction, which means we go from reactants to products. Now, sometimes in some chemical reactions, we may have arrows pointing in both directions, which would indicate that it's at equilibrium and reactants and products are being formed and broken down at an equal rate. And most of the examples you're going to see here today, that is not what we're going to be looking at specifically, okay? So reactants versus products, that would be good certainly to understand. When we look at chemical reactions, we know all chemical reactions will involve a change in energy, and that change in energy often comes from the bond energies that we see in our reactants versus those bond energies that we see in our products. And we're going to look at bond energy. I'm just checking over here on the next slide, okay? But the energy in a bond is essentially what we need in order to maintain that molecule, right? To maintain the stability there. When we need to initiate a reaction in a cell, in the body, in any living thing, we always have to invest a small amount of energy to kind of kickstart the process, right? So when we go through this process of doing a chemical reaction, we always invest energy in the form of what we call activation energy. And that is a vocab term, so certainly make sure you get that. Activation energy is the amount of energy we need to invest or absorb in order to start a reaction. Okay, so if you're rolling a boulder down a hill, and you're going to see this example, I think, No, you don't have this example in this one. Usually I use this as an example. If you're going to roll a boulder down a hill, which I don't know why anybody would try to do that, but let's say you're sitting at the top of a hill and you want to roll it down. Well, you can't just let it sit there. It's not going to roll on its own. You have to give it a little bit of a push, right? So that's the idea behind activation energy. The diagrams you see at the bottom here, you do not need to write down. I'm going to have you actually jot those down here on the next slide. I've got another couple of graphs that look very similar with some more labels. Okay. So here is representing our activation energy in this type of chemical reaction. And it has to do with my reactants, where we're starting, and then where we need to end up in our products. Activation energy on this one over here on the right, you can see is much shorter. That's because my reactants start out higher, my products end up lower. And this is what we call a chemical reaction that's going to release a good amount of energy. In contrast to that, this one absorbs a lot of energy because I have products that have higher energies than my reactants first started with. Okay, so I mentioned bond energy on the last slide. So bond energy is the amount of energy we need to break or form bonds between any pair of atoms in a molecule. Basically, what we need to either break down a molecule or to form a molecule. The higher the bond energy, the further up we're going to place that energy on this scale on our y-axis. So these two graphs you are going to want to jot down, okay? So our y-axis in both represents increasing energy. The x-axis in both is representing our reaction progress. So in other words, we're going to always have our reactants on the left, just like we would write them in any chemical equation, and our products are on the right, right? So when we do this, wherever my reactants fall and wherever my products fall is based on the amount of energy we think it would take to break or form those bonds. And this is certainly something that would just be given to you, right? This is not something that I would ask you kind of to know off the top of your head. But based on where these things fall, you should be able to predict for me what type of reaction it is that we're looking at. Is it going to absorb more energy? Is it going to release energy? What is that going to look like? So in this scenario here, I have an average energy of reactants that's higher up. My activation energy is small, right? Because I only have this small hill to climb. And my products energy is going to be way down here, okay? So what this means is that in this chemical reaction, actually, I'm releasing the difference between my reactants and my products. That energy is being released, okay, as part of this process. On the other hand, in this one, it's kind of the opposite. I start out with reactants with very low bond energies, and I need to create products here that have much higher bond energies. And so to do that, that requires a whole lot more activation energy, right? You can see it labeled right here. This huge gray arrow is the amount of activation energy. It's significantly different from the panel on the left. All right, and then the reaction starts and kind of rolls down the hill, right? And then we end up with our products here with a net gain of energy. We've absorbed or added energy in order to do this. What we know about these bond energies, we tend to add energy in order to break bonds, and we tend to release energy when bonds are formed. So that kind of drives the distinction between this. It's not that this is just forming bonds and this one's just breaking bonds. That's not what I'm saying, but I'm saying kind of that net gain of energy. The energy that's absorbed is being done so in order to create these higher energy molecules, right? There's something going on there that's driving that process. So we can distinguish between these two graphs with two additional terms. So whether a reaction releases or absorbs energy, again, depends on bond energies. You should have that from the slide before. And then these two are new. So you're going to want to make sure that you add those two bullets for me because the terms that you see there are certainly going to be important. So we have what we call exothermic reactions and we have endothermic reactions. And if you remember your Latin roots, exo means out or outside, and endo means inside or within. So when we talk about exothermic, we're releasing energy to the outside. Okay, this is therm meaning heat. A lot of times this energy is generated as heat. And so that heat energy is being pushed to the outside. All right, that's the idea. For an endothermic reaction, we're absorbing that heat. We're absorbing that energy inside or within the molecule. So they're actually going to absorb energy in the scenario where we're looking at these graphs. So the one here on the left, as you draw that, we're going to label that one as exothermic, and the one on the right here can be labeled as endothermic. And the main reason for that is what we're seeing right here. So this arrow here is representing energy absorbed. It's a difference between my reactants and my products. This arrow here is not energy absorbed, though. This is representing energy released. Okay, so again, that can be in the form of heat. That can be in the form of light. That can be in the form of different forms of energy, right? It doesn't have to just be heat. But when we talk about our cells and what's happening in a living thing, it tends to mostly be heat, which is why it's so important that we have so much water in our bodies and in our cells to help with that concept of heat capacity and high specific heat, and the fact that water functions really well to kind of resist changes in temperature, like we talked about back in section 2 too. So if we have exothermic reactions where we're releasing a lot of energy, we want to be able to ensure that our cells aren't also changing temperature as a result of that. Okay, because that energy has to go somewhere. We just need to ensure that it's not impacting the whole living thing. Okay. So I will say for this, you will see several of these on your test. I would definitely be familiar with the structure of these types of graphs. And I would be familiar enough that you could actually even label it if I asked you to label it, okay? But certainly understanding which one's exo, which one's endo, and why we call them those terms would definitely be important for this test, okay? So make sure you've got that for me. All right, the last thing in chapter 2 here is to look at enzymes specifically. So enzymes are specific to a type of chemical reaction, and these are also only found in living things. So notice I'm using the word up here, biological. Oh, too far. Let me scoot back. I'm using the word biological here, and what I'm trying to represent with that term is to emphasize to you that this is only in living things. Enzymes don't occur naturally in the non-living world. This is specific to cells. So biological reactions will often need catalysts, and they need catalysts to help them kind of speed up their chemical reactions. And enzymes are a type of catalyst that we see in living things. So if you've ever heard the term catalyst before, if you've ever done chemistry before in any other type of science class, you might have used a catalyst to speed up a chemical reaction as it happened in the classroom on a lab bench, okay? So enzymes function in much the same way. The difference, again, is that they're specific to cells, and there's many different types of enzymes, each of which catalyzes its own specific chemical reaction. What they do in order to speed up a chemical reaction is not really anything involving speed. The speeding up of the reaction is kind of an indirect effect in a way. What they actually do is they lower the activation energy. So remember, the energy that we have to invest in order to get the chemical reaction started. And if we lower that activation energy, we in turn are going to kind of inadvertently speed up the chemical reaction because we make it easier to push that boulder up the hill. We make it easier to get the reaction started. Okay, so if you look at the graph here on this slide, and this would be something you could add to the graphs that you just drew, add yourself a little enzyme line. The enzyme line here is represented in red, and as you can see, we started out with a reaction pathway without an enzyme up here that was much higher in terms of its activation energy. When we added an enzyme, it lowered the height of this hill, right? It dropped it down, so now my activation energy is much more shallow. There's a lot less that I actually have to invest. Notice I didn't change anything here with my reactants or my products. My bond energies are still exactly the same. So an enzyme's only true role, its only function, is to lower the activation energy. Okay, it does not alter reactants, it does not alter products, it does not alter the direction of the reaction. All right, so they're important in a lot of ways just because in our cells, these chemical reactions don't happen quickly enough on their own. So they need the help of these enzymes to kind of get them started and lower that activation energy, okay? Now, how enzymes actually do this is through something called a lock and key mechanism, which is kind of an odd name maybe, but I like it because it helps me remember exactly what's happening here. So we know that an enzyme, typically these things are proteins, and proteins, like we talked about in section 2.3, are made up of monomers called amino acids. Those things link together with peptide bonds, and then they fold and form these unique kind of structures. We saw that with hemoglobin, with all the different polypeptide chains that interact and fold and kind of twist. So enzyme shape is designed to accommodate specific reactants and products, which means that an enzyme can't just catalyze any random chemical reaction. It's specific for one type. All right, so the shape and the structure determines what can bind to it, and it's so specific, in fact, it's related to this idea of a lock and a key. So I always use the example of at your house. If you have a key to the front door at your house, you can make many copies of that key, right? You could also have the same lock there, right? You could buy many copies of that same lock and use them on different doors in your house. But the key, for lack of a better word, right? The important part about this is that we couldn't just take any key and use it on that lock, right? It has to be the one that was cut specifically to fit the lock on that door. So where we can make many copies of reactants, we can have many of the same products, we can have many of the same enzymes even. But what matters is how these things fit together. They have to fit together perfectly, and if they don't, there's no chemical reaction that can occur. All right, so the reactants that bind with enzymes in this lock and key mechanism are called substrates. And that's just another like biochemical term for reactant. It means exactly the same thing. So if we have a substrate, like in this scenario here, like sucrose, remember this is what we just call sugar. This is what you buy in the store as sugar. It's what you consume if you add sugar to anything you're baking and cooking. So sucrose here is going to be my reactant. This is going to be a reaction here, if you kind of look down the line, where we're breaking this bond to create our monomer subunits. This is my disaccharide. These are my monomers, glucose and fructose. So I have an enzyme here that contains what we call an active site, which is the exact location where these reactants or substrates will come in and bind. And you can see my substrate looks like it should fit perfectly here, right? So the substrate floating around in the cell and the enzyme floating around in the cell, they find each other. Miraculously, they find each other. And here is where they start to interact. And as they interact, we're going to catalyze the formation of this chemical reaction, which in this case is breaking a bond. The way that that's done is through a process called hydrolysis, which I don't have any more on this, but it's essentially using water to lyse or break something, right? Lyse was a Latin root that you all looked at, split, loose, and break. So we're using water to break that bond there. So the bond breaks, we release our two products here, our two monosaccharides, and notice my enzyme is basically unchanged. So this means that a single enzyme can be used for multiple reactions, and that's really beneficial in a cell where we're kind of limited in terms of what we can actually produce. We don't want to overproduce anything. We want to make sure that we're using all these things to the best of their abilities so that we get the most out of these molecules that take so long to create, okay? So the last example here's another example of another enzyme kind of substrate complex, lock and key. So we have our reactants here, water and carbon dioxide. They bind to the enzyme, and notice this one's probably actually a little bit more realistic. Active site is specific for these things, but notice how this protein is actually kind of shaped. It's not as simple as the one we saw on the last slide. So CO2 and water come in and bind. They form this enzyme substrate complex. The substrates are converted here. All right, so they're actually going to be combined, and they form this thing called carbonic acid. If you remember, carbonic acid is the thing that actually gets formed in oceans through that process called ocean acidification, and that can be kind of problematic because that's where CO2 is just kind of readily interacting with water on its own without the help of these enzymes. And dissolving in those water ecosystems and lowering the pH, which then threatens all the living things found in those places. So this is kind of that process, but as it would actually happen in a living thing in a cell. Okay, again, product gets released, and notice my enzyme looks exactly the same. So again, we can reuse the enzyme, we can go on and use a product for whatever it needs to be used for, and we've catalyzed this reaction in a much simpler way than would have happened without the help of an enzyme. Okay, so again, enzymes, their major role, speeding up chemical reactions by lowering the activation energy and essentially providing a place for these reactants or substrates to come in and interact. Okay, so that is it for chapter 2. If you've got questions, of course, let me know, but otherwise make sure you're reviewing this material for your test coming up because it's going to be a big one for sure. All right, so other than that, that's all I've got for today. So take care, and I will see y'all next time. Bye.